Esposito. I'm the Curator of Arachnology at the California Academy of Sciences. And in front of me today I have a jar of scorpions. And all these scorpions belong to the same genus. It's called Centauroides, or the bark scorpions. But in particular, I have one little vial in here that I'm super excited about, because for me it has like a great deal of history. It's actually the first vial of specimens that I ever borrowed from the Academy's collection. The Academy research collections really represent a lending library, and we loan those out to people, researchers all over the world. And when I loaned these specimens, I was a PhD student at the American Museum of Natural History in New York, and I was studying this group of scorpions. And these specimens in particular are from the Galapagos Islands. And they're kind of an enigmatic species because we don't entirely know what their closest relative is, meaning we don't know where they came from before they got to the Galapagos. And so I was loaning them to extract some DNA. And when I attempted this, the specimens had not been collected for DNA. Like, they were just collected because somebody was there and was trying to improve a scientific research collection, never thinking that DNA could be taken out of one of the things that they were collecting at the time.
When were they collected?
These ones were collected. There's actually like a tiny label in the bottom, and that's another great thing about collections, is we can tell everything about every single specimen, including where and when it was collected and by whom. And it tells us all that information, usually on a tiny, tiny little piece of paper. And so these really represent a place in time and what was happening in that environment at that moment. What that means is, you see that here it says 1971. These were determined by this gentleman, Kinzelbach, in 1971, but the species was originally discovered in I think the 1920s. It was the first time it was formally described. And these specimens were collected in 1954. So it took 20 years before somebody even came along and told us what they were, what we were holding here in our collection. And it took another 40 years before somebody, me, a young PhD student, came along and wanted to do research on these specimens. So it just goes to show that things we collect today, we have no idea what the future of them is going to hold and how they're going to contribute to research.
Right, and as time went by, they actually got more valuable.
As time went by, they got immensely valuable, because we'd prefer to take things out of collections rather than to go into the wild and remove them. Mm-hmm. And so these have immense value. We can take DNA out of them. In some cases, we can look at the isotopes and try to figure out what they were eating, or whether there were anything like a nuclear reaction occurring during their life or before their life. And so we can gain all kinds of information just from these now 60-some-odd-year-old specimens, going on 70.
And Lauren, it's so cool that these were the first you borrowed, and now you're here and actually adding to collections.
I know, it's amazing. And I really hope one day I get to go back to the Galapagos and study how this population has evolved through time. And I could do that because I have these specimens.
I'm Bart Shepherd. I'm Senior Director of Steinhart Aquarium here at the California Academy of Sciences. And this is a tiny little fish. It's called Plectrantheus ahi-ahi-ata. And it is a fish that I found and collected on Rapa Nui, or Easter Island, a year ago in the spring. This was my first species description, where I was the senior author and actually got to select the name and everything for the fish.
And this was quite recent, right?
Yeah, this was just about a year ago, a little bit more than a year ago, and we were doing a deep reef exploration, looking at the Twilight Zone reefs in Easter Island, which is part of our Hope for Reefs initiative here at the Academy. And we had been there 4 or 5 days and done a bunch of dives and really hadn't seen anything new. And it was my last dive, and come to find out, we did this dive, found a rock in the middle of a big sandy area, and I think we got 4 or 5 new species of fish on that one rock and one dive. So pretty spectacular day for us.
What does it make you feel, just as a scientist and explorer, that you can still find new species?
I think that's one of the most amazing things about my job, is that we live in this planet where everything is known and we're all hyper connected, but you can still go and find places that nobody's ever seen before, and go and visit places that no person has ever set eyes on, and find animals and plants and things that nobody has ever seen before. Our Hope for Reefs initiative really has sort of four main buckets, and it's all looking at addressing the main causes for coral reef decline. And so there are scientific aspects of that, including some deep reef exploration and looking at these unexplored coral reefs in the twilight zone. There's a coral restoration component, which is advancing the science that's needed to actually restore coral reefs and put healthy corals back on degraded reefs. And then there's public engagement and outreach components here at the Academy and digitally through immersive media like our planetarium show, Expedition Reef, that we just opened this past year.
Yeah, neat. And I understand you have another specimen as well that you've pulled.
Sure, I have another one. This is the Caribbean elkhorn coral, Acropora palmata. And this one is particularly important to me because I can remember growing up and visiting reefs in Florida and in the Caribbean as a kid and on spring break with my family and snorkeling and seeing these huge fields of elkhorn coral out there. And in the 70s and 80s, really, this species of coral, which is one of the most dominant corals in the Atlantic, it suffered from huge declines from disease, and it went down to about 90-95% decline. So the historic populations are basically gone. But as part of our Hope for Reefs initiative and working on coral restoration, this is one of our focuses of activity. So we're looking at how we get out into the wild and we can restore populations of these on reefs throughout the tropical Atlantic.
So it sounds like a specimen like that is both a record and kind of a reminder of what you're working for.
Yeah, exactly. I mean, this thing is sort of like a talisman for me to keep my mind on, the fact that it's a critically endangered species and that it, without anybody caring and trying to make a difference, it will just vanish and disappear and be gone forever.
So my name is Pim Bongaerts, and I'm a curator here at Invertebrates. So I focus on tropical coral reef corals. And so I brought one.
Yeah, let's see it.
And this is a very special specimen. It might not look very special because it's not particularly beautiful or big or anything like that. But it's actually a very important specimen because it's the deepest reef coral that's ever been collected from the Great Barrier Reef. At a depth of 125 meters or 410 feet. And so for a long time we didn't actually know how deep corals that are light dependent, that still depend on photosynthesis, extend down the reef slope. And so this is one of the first physical records that prove that they can occur all the way down to those depths.
And why is it important to know where corals occur?
It's important to know because obviously if we're designing marine protected areas, we need to know where in that large area the corals actually occur. For a long time, we've just been looking at shallow reef corals in the Great Barrier Reef. And so most studies are done in 20 meters, 60 feet or shallower. But it turns out that there's this incredibly large area of deep reefs. And we estimate that it might be actually equivalent to that of shallow coral reefs on the Great Barrier Reef. So there's this whole second Great Barrier Reef. And we obviously need to know what's there and where it occurs to be able to protect it. I didn't actually collect this specimen, but this little guy did. So we call him Kermit. Because of his color, obviously. And so Kermit and his little friend, because we've got another one, the same color, were actually the ones that brought up this specimen.
Hi, I'm Nathalie Nangalingum and I'm a curator of botany. And this is the specimen that I chose. It's a specimen collected in the late 1800s. And if you look over here, this is the date where it was collected, 1899. And it was collected all the way out in Kings Canyon. So right now it takes 4 hours to drive there. But if you think of 1899, Alice Eastwood, who collected it, she would have had to take probably like stagecoaches and horses, and so it really was quite an expedition for her to get there. And so this is a new species that she discovered, and these are the plants that she used as an example of those new species. And what I find is really special about these plants is that Alice Eastwood actually went into the Cull Academy after the 1906 earthquake, and she saved this specimen and about 2,000 other specimens from the fire that then swept through all of San Francisco. And so this is really special for us. We keep it behind locked doors, and so it represents the first time that anybody ever recognised this species and wrote a scientific name for it. And so this is the scientific name, Delphinium polycladon. And Alice Eastwood, you can see she is the first person to name it, and so it's given her name afterwards. Alice Eastwood raced to the Cull Academy to save these specimens. And so what she did was she found that the stairs had crumbled, and so she climbed the staircase to get to the 6th floor and then bundled all of these specimens and used ropes to lower all of them down and saved them from the fire. And so the Cull Academy Herbarium, which probably had about 200,000 specimens, was all burned, and we were just left with 2,000. And so Alice Eastwood then, she had no job for a while, but her job was then to really expand the herbarium, and she came back to Cull Academy and started collecting many more plants after our tragedy.